Hello and welcome to the 1516 Biology Curriculum Overview for Unit 6, Cellular Processes of Cell Growth and Differentiation. As we look at our ISD, we notice that TRS suggested a duration of 11 days. However, on our GPISD YAG, we have exactly 12 days starting on November 5th, running all the way through November 20th. Here you have our processing skills that are in black, and the supporting are green, and the readiness are in red. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have exactly 9 SEs that are embedded throughout Unit 6, and majority of those SEs have been covered already from previous units. However, they will be spiraled back in and used in different contexts as it relates to this unit. And a few notations again, looking at our prior to the unit, we want to make sure that we're using our prior to the unit to engage the students and access that prior knowledge. This is prior knowledge that they should have from biology, previous concepts that we've covered, as well as in 7th grade when they've gone over genetic material found in genes and the chromosomes and inherited traits. So this is the information that you want to definitely use to develop engaging activities to start your unit. During this unit, of course, we use this information for our explore activities, different ways for the students to explain their understanding of the unit, for coming up with activities for the elaborate, and to gauge what the students understand and prepare them for our PA, which is the evaluate. Now, after this unit, the students will be able to take this information that they've gathered in Unit 6 and apply it in further deeper terms of genetics, evolution, and viruses and bacteria. So we want to make sure that we're using all of our tools to make sure that our students are successful in Unit 6 so that we can build on this knowledge. Now, again, as I mentioned, we have a number of SEs that will be embedded throughout this unit, and I have the data from our district assessments from 13-14 Q2 summative as well as the 14-15 Q2 summatives, and I have highlighted in red the readiness standards and a couple of the supporting standards. These are the main standards for this unit. However, the others that are in green are also spiraled in and have been covered in previous units. So I just wanted to make sure you understood the differentiation between the two and the colors. And as you can see, we have demonstrated some growth from 13 and 14 on a few of the SEs. We've fallen down in some and we've increased in others. So let's make sure that we're using our district data to gauge where we are and where we have been as to understanding where we should be going and getting that plus 10 as we continue on in the 15-16 year. And briefly, as you look at how the SEs were assessed on the STAR assessment from 2012 all the way to 2015, you'll notice that we have demonstrated growth each year in our supporting standards for the most part. For A, we went from a 32 to a 30 to a 69. It was not assessed in 14, but it was assessed in the 2015 assessment. As well as in 5C, we saw an increase. In 5B, there has been a significant increase. So in our supporting standards, we are seeing an increase in growth being demonstrated amongst our students on those specific SEs. However, in our looking at our readiness standards, there is a bit of a stagnation going on, and so we want to make sure that we are definitely covering those readiness standards in depth and providing critical thinking, and make sure we're using processing skills as we develop our lessons and activities to address these standards. We want to continue to see growth amongst all of our standards, but let's make sure that we are definitely focusing on these college readiness standards. Now, as I go through the STAR assessments to understand how the SEs, both supporting and readiness, have been assessed on the STAR assessment, The purpose of this is to understand how the state interprets the SEs. Understanding how the state interprets the SEs helps you to better prepare your students and develop effective 5E lesson plans that will prepare the students for the STAR assessments. The questions are rigorous. The questions are involving processing skills. Some do not. However, the students must be able to read the question, understand what the question is actually asking them, and pinpoint the best answer to address that question. And so that's the purpose of us going through these different SEs and seeing how they're being interpreted on the state assessment. Now, as I go through the readiness standards, I have 3 questions in here, one from each year, 13, 14, and 15. And on the supporting standard, I only have 1 question, and it will come from either one of the years, either 2015, 2014, and 2013. Again, you can retrieve these questions from the Leap Forward STAR Release Assessment for 15-16. And so looking at B5A, we're focusing on describing the stages of the cell cycle, including deoxyribonucleic acid, DNA, replication and mitosis, and the importance of the cell cycle to the growth of organisms. And as we see here, this is a readiness standard, and it involves utilizing the processing skill 2G, where they have to interpret a graph or some type of data. And the model here represents the change. The question is, the model represents the change in DNA content of a cell during the cell cycle. Which part of the model represents the S phase? And so they have to understand, well, what's happening in the S phase? We know that DNA is replicating. And so if DNA is replicating, that means there must be an increase in the DNA. So they must be able to interpret this model and say, okay, here I can see there is a growth or increase in this data table, so section 2 has to be S phase. Okay, and so as we can see, the correct answer was G, and G is 2. So that's just a different example of how to incorporate interpreting graphs in the context of what's being asked in the SE. Different ways to put that into your lessons so that when the students see any type of model or any type of data table, they know how to approach it, and they know how to use those same processing skills they've used during class and during lesson activities and apply it and answer the question. So that's the whole purpose. We are not teaching to the test. We are teaching the SEs and understanding how the state interprets the test. Interprets the SEs. Thank you. So again, we're looking at B5A. We have a 2014 question where you have a model of a cell cycle, stages of the cell cycle, and again, please make sure that the STAR A accommodations are being used for the students who will have STAR A accommodations. They can be used as a tool or graphic organizer strategy for all of your students. However, it would only be available for those who receive that STAR A accommodation on the test day, as well as during the district assessments. Now, on this 2013 question, we are looking at a diagram or a picture that shows the cell cycle in a different way. So there are multiple ways of viewing the stages of the cell cycle, and the students will have to interpret which phase of the cell cycle is shown here in the image. And of course, this one is anaphase, and it would be H. And you can see that the students in the past have had some difficulty identifying the difference between G, which is metaphase, anaphase, and telophase, and also prophase. So we want to make sure that we're doing different various activities, and the students are seeing it in different ways, not just one way, because they can see it in different ways on the assessment. So we want to give them different opportunities to see different visuals and different models of the cell cycle. Now looking at our supporting standard, again, there is only one question in this presentation over 5B. We're looking and examining specialized cells, including roots, stems, and leaves of plants and animal cells such as blood, muscle, and epithelium. And so the basis of this type of question, again, you're using processing skills to understand or demonstrate an understanding of this supporting standard. The two most abundant types of white blood cells may both be involved in a response to which of these. And so they have to analyze and break down this graph, looking at the percentage of the total white blood cell count. Look at the two most abundant, which are the lymphocytes and the neutrophils, and look at their function. And based off their function, they need to determine which of these responses will be involved, or the functions of these two types of white blood cells will be a response to which of these injuries or infections, what may have you. And so, of course, the answer here was in a bacterial infection. Okay, so again, I'm not going to go through each of these questions. I just want to give you a quick glance at the different types of assessment questions over the SEs. And we're here looking at B5C, which is I will describe the roles of DNA, RNA, and environmental factors of cell differentiation. And this is a question that they'll see, and again, they're using processing skills. And so this, you can read it on your own and go through and look at it in more detail. We have a 2015 question over 5D, another supporting standard. Recognize the disruption of the cell cycle that lead to diseases such as cancer. On 6A, our readiness standard, of course, there are going to be 3 questions in here from 15, 14, and 13. Here is a segment of DNA represented in this illustration. How is the information for a specific protein carried on a DNA molecule? And this question is addressing, again, 5A, where they would have to identify components of DNA and describe how much information for specifying the traceable organism is carried in the DNA. And 2014 question over 5A, another model using another processing skill. And we have another model using a processing skill 2H, a model of DNA, where the students will have to answer the question and understand what this is representing, what the arrow is representing. Now looking at 6B, recognize that the components that make up the genetic code are common to all organisms, and there are so many different ways that you can incorporate making sure the students understand that the components that make up all organisms is universal. It is universal. They contain all of the same components: a phosphate sugar base, phosphate sugar base, phosphate sugar base, the same for nitrogenous bases, but the difference is the sequences of those bases. Now, as we go more into depth, we're going to look at the IFD PAs and our modified Unit 6 PAs. Again, last school year, the biology department met through our curriculum overviews, and we modified the instructional focus document PAs because most of them were not the best form of delivery for the students to demonstrate their understanding. So we are making sure that each of our campuses know, our biology campuses know, that we are utilizing 14-15 modified PAs throughout the entire year. And so we have 2 PAs for this unit. So make sure that you split your PAs up over the number of days you have allotted for this unit, which is 12 days. And modified PA1 is here, label and describe the steps of DNA replication. Use the model to explain the following: how new DNA is synthesized and the general differences between this process in prokaryotic and eukaryotic cells, the significance of DNA replication, the commonalities of the genetic code in all organisms, and the limitations of the model. And so that's all for Unit 6 PA1. And you have Unit 6 PA2 that I allow for you to read at your own time and discretion. But I just want to emphasize again, we are utilizing the GPISD 1415 modified PAs, and you can find them on the secondary shifted resources Dropbox. Okay, and so we're looking at the modified PAs, and we're chunking the SEs that are to go with each of those PAs. And so here you have your specificities, and I'll just put an SP. So, and you have your SEs here. And all I did for this one was chunk the readiness standards because I had limited space, but of course you would also embed the readiness as well as the supporting standards that go along with them for each of those PAs. So here we see B4B, which is a readiness standard for the modified Unit 6 PA1, as well as B6A and B5A. And B5A will also be embedded in the Unit 6 PA2. So this is just a quick snapshot and glance of the chunking of the unit PAs to their standards and specificities. So our essential questions, again, our essential questions, and there are sample guiding questions in the IFD and available on TRS for the unit, so you don't have to try to figure out or come up with any questions. They are already there and available for you. Here you have the essential questions again. The first two are specifically for Unit 6 PA1, and that is what is the importance of DNA replication to the growth of organism, and what is the importance of each of the cell cycles, of each of the stages of the cell cycle to the growth of an organism. So both of these go directly with PA1, and the other questions in addition to questions 1 and 2 all go with PA2. So here are your available PA essential questions that you can utilize in your explain portion. You can utilize them in your elaborate. You can utilize them in your evaluate. You can utilize them all throughout the lesson, but you want to make sure you're checking for student understanding. And again, the best way to do this, I believe in my lessons, I would use them in the explain portion as well as the elaborate. If you have any questions or you need help with that, please let me know. Okay, real quickly, we have our misconceptions as well as our vocabulary, our key content vocabulary, as well as related vocabulary. And again, I don't put all the related vocabulary because there are so many tons and tons of related vocabulary that it doesn't all fit here. But a quick snapshot, please make sure that we are addressing these misconceptions. And if you look back at our data and look at the misconceptions, you can see the trends and the connections that lie between the misconceptions here and what the scoring is on our data. So you can see if the students really understood that concept or that SE based off of what misconception they may still have or they may have had at the time that they took the assessment. One of the biggest misconceptions I know from my own experience is students think that all organisms have a different genetic code, but the truth is the genetic code is the same. The components are the same. Let me correct myself. The components are the same. However, the sequencing of the DNA is what has changed. So we want to make sure that they understand that specifically that the components of DNA is universal. And you can go through and read the other misconceptions, and you'll want to create activities that will address those misconceptions and see what the students already know in their engage about these different misconceptions or what they think they know, and develop explore activities that will help them correct their understanding and see the truth behind what their misconception was of that SE. Now, key content vocabulary, we have cancer, we have cell cycle, cell differentiation, DNA replication, epigenetics, gene, mitosis, replication, sort. Those are key content vocabulary terms. We want to make sure we're always using those and spiraling those in throughout the lesson. Some of these words they have seen before from 7th grade, so we want to make sure we're throwing those words back in and having them give us the words and give us their definition of what they think it means and put it into their own words. So we have tons and tons of vocabulary and different ways that you can use this information. Okay, and before I wrap up, I want to cover over one of the main focuses of this year is making sure that we are implementing writing across the curriculum. And for science, secondary science specifically, we are using CERs, which is claims, evidence, and reasoning. And we have spoken with the English, secondary English department, middle school and high school. And the CER rubric that we have for science is in alignment with the SAQ rubric that the students have for English 1 and 2. So I have provided these rubrics on the Dropbox, and they are available for you to read through them. They are available for you to compare, as well as allow the students to have a copy of both where they can see the relevance and how they're aligned across the curriculum in both English and science. Sometimes you get the question, what does English have to do with science? Well, they need to understand the connection and why you need to be able to write in science effectively the same as you write in English effectively. So claims, evidence, and reasoning is our form of writing across the curriculum that we're using for the district. Okay, and you may say, well, how do you structure a really good scientific explanation, or how do you structure a CER explanation? Well, the explanation should consist of 5 to 7 sentences, which is a complete paragraph, a full paragraph, anywhere from 5 to 7 sentences. And your claim is the answer to the question or the answer to the prompt. It's what do you know? It's the answer that you're giving to answer that thing. This is my claim. And when the students write their claim, you want to make sure they're not saying I think or writing in first person period. I think, I believe, I hope, I know that, because I wish upon a star. I don't know. But you want to make sure that they are using proper terms and just stating the exact claim. The bird flew out the sky and hit the ground and died. That was my claim. Now I need to provide evidence to back up how I know that the bird flew out the sky, hit the ground, and died. And so my evidence, my claim, I would have one completely structured sentence that states my answer to that question or prompt. The evidence, 2 to 3 completely structured sentences with three pieces of data, facts, or text evidence that supports the claim. This is very important because you can pull evidence that's irrelevant to the claim. So we want to make sure the students are pulling evidence that is relevant to the claim and it is supporting the claim. In your reasoning, this is the part that confuses a lot of people, even adults, even adult teachers. It confused me at one point as well. So your reasoning is what connects this piece of evidence to my claim. How does my evidence support my claim? And so when you write your reasoning should be 2 to 3 completely structured sentences, and each piece of evidence has to be explained as how it's relevant to the claim. And so I provided an example that I pulled from an article I read online, and it was a good example of how it should be written in terms of pulling the evidence and connecting that to your reasoning. And it reads, write an explanation that states whether any of the liquids are the same substance. Liquid 1 and 4 are indeed the same substance. Looking at this data, the properties include density, color, and melting point. Mass is not a property. Density, color, and melting point are all the same for liquid 1 and 4. Since all of these properties are the same, 1 and 4 are the same substance. So that was a very thorough, straight to the point. The student didn't say, I think, I wish, I knew. They just stated their claim. Liquid 1 and 4 are indeed the same substance. They provided their evidence from their data, and they provided the reasoning that connected their evidence to that liquid 1 and 4, which was their claim. So here's an example, and there are several other examples. A good way to help your students understand or get more acclimated to writing effective CERs is to practice it with them first. Don't just throw it into their face and say, hey, write a CER, and they have no clue really what it should look like. So you want to model that for them. So I've provided you with a CER PowerPoint. Again, that's available on the Secondary Shifted Resources Dropbox. And I would be more than welcome. I will be more than happy to come by and work with your PLC and show you how to utilize that and go through it with you and your PLC or individually, so that you can effectively use it in your class. And you may wonder, when can I use this? Well, I can use it to have students write their response to a scenario, or I can ask an open-ended question, or I can take a STAR question and I can take away the multiple choice and say, hey, answer this question, analyze this graph, provide me your claim, your evidence, and your reasoning. As well as when the students write formal lab reports, it is their conclusion. So once you get the hang of writing effective CER, you can write. You can write in science, you can explain your thought directly what it is, provide your evidence, state it, and tell why. And that's where we want to get our students to in biology, and make sure that they're doing this so that it can carry on into higher levels of science as they go through high school. If you have any questions over this, please let me know. And at the conclusion of our presentation, last but not least, a few important reminders. The STAR A accommodations I mentioned before throughout the presentation are available on the Discovery Ed Board Builder. They're under the Assessment and Research folder under District Content, and you can click on that and go to Science and Biology, and all of the accommodations will come up. Again, you can use those accommodations in your class for all your students. However, on district assessments, as well as the STAR assessment, only those students who receive STAR accommodations will be provided that graphic organizer or mnemonic. We have SST, Strategic Science Talk, that's available for everyone to join in and ask questions, and whatever you need, just come. That's our means of communication for science, secondary science 6 through 12. The Edmodo group code, joint code is TU4HFR. And again, everything that I have gone over through the curriculum overviews, exemplar lessons, CERs, rubrics, SAQ rubrics, they are available for you, PowerPoints, on the Secondary Shifted Resources Dropbox. I hope that this curriculum overview has been beneficial and that it will help you. If you have any questions, if you need further information, please do not hesitate to contact me. You can email me directly, erica.ingram@gpisd.org, or call me at extension 5351. I hope that you have found this to be useful, and have a great day. Thank you.